So okay, let's just give it a couple more minutes, maybe 1 or 2 minutes, and then we'll start the second. One of the important part of the presentation, obviously, is the data wrangling and all the necessary steps we need to perform or understand, in terms of before I actually start building the model. And it's one of the important aspects in any analysis to understand and clean the data and make sure that the data is coming up in the appropriate format, because format itself will define and provide some sort of set some limitation towards the method you can apply to the particular data. You could have a combination of a categorical data, you could have data raw data, let's say a number of counts of the particular from the particular NGS analysis. And the question would be like, how can you, for example, merge together different modalities or different type of data into one of the big framework? And there is no unique solution, obviously, in this case, but before you actually start combining and building them into one framework, it's important that you clean up and you don't supply any not so well posed samples. And this is going to be the focus of that second half. All right, let's move on. So what we're going to talk during this part of the training is why do we need to do data processing, data cleaning, obviously data integration and transformation, data reduction, discretization, concept of heuristic generation, and then we'll summarize in a few points. Again, this is a very important field. There is a lot of discussion can be done here. For the sake of the time, we're just going to be very basic, maybe just scratching the surface. But I think it's important to scratch that surface. And if you would join us for the 3-day training, commercial training activities, then obviously we'll spend at least half a day on some of the examples and then trying to understand what is the impact of doing the appropriate or different data cleaning process, and then how can you actually enhance the cleaning process, not just simply deleting bad data points, because sometimes if you delete just all the data bad data points, you will end up in a very small data set, and then it leads to the next problem, like you don't have a sufficient enough data points to make the statistically significant claim about your built machine learning model. So they're all interconnected, and their life is obviously not perfect. Most of the time, you don't have a perfect data coming up from your experimental campaign, and that's very important to be open-minded and just don't throw out the baby out of the bath before actually analyzing and making conclusion where you can adjust the data or not. Anyway, enough about the summary. Let's move on to the first question. Why do we need data processing? Obviously, the data in the real world is dirty. It's sometimes incomplete, noisy. Noisy could be errors which is coming from the out instrument, or is it environmental noise coming up? It could be also biological variability, meaning that it will provide some of the distribution across different replicates in your experimental campaign. And some of the data could be inconsistent, especially when you're trying to combine different modalities. You would see that there is some observation which is kind of contradict to each other to some extent. Of course, the first reaction would be just to remove all those data points which incomplete, noisy, inconsistent, but then you may end up in a situation of having no data at all for your training or very limited training data set. So therefore, it's actually one of the parts of the machine learning to be able to find a way to incorporate the data and still learn from the data. And biology and biotech itself is actually very different compared to any other industries. For example, automotive, if you're talking about the self-driven car, the controls of the data and the noisiness and inconsistency is much easier compared to the biological system. And just because of the fact that what you can measure and the biological variability aspects, across different human being or different, species of your interest. So therefore, it's even more tricky and more important than biotech or biology domain, computational biology domain to tackle those problems versus, let's say, aerospace industry or self-driven car automotive industry. For them, it's very important to build the accurate model, but the data accuracy and completeness and noisiness is probably less drastic compared to what we can measure and what kind of data we are getting. So obviously, the next point of course, quality. Quality of your mining or quality of your results, and always will depends on the quality of your data. Garbage in, garbage out, that's a universal statement. Obviously, therefore, you need to make sure that whatever you're using has a reasonable quality, or at least you understand what is the uncertainty, and you propagate that uncertainty in your data towards the uncertainty of your prediction. That's a very important step, and therefore, that will allow you to properly utilize some of the issues mentioned in the previous section. Obviously, when, as I mentioned already, you have multidimensional data, even like multimodality data, which comes from the different aspects. It could be, let's say, combination of your next generation sequence data and then clinical records. It could be just, I don't know, some of the single cell data, and then also some of the data about the chemical compound use. So it's very important that, you still, worry about the data quality. You understand all the limitations. If there is certain uncertainty in some of the parameters, the information, it has to be propagated, it has to be taken into account, and then results has to be a reflection of what the uncertainty was in the data, which should result in the uncertainty of the results. If you provide your results and estimate with the correct uncertainty or error bar or probability of the belief, it still could be very valuable because it allows you still to make certain statistically significant claims, and therefore don't be just scared by the fact that, oh, I don't have enough data, or some of the data are really inconsistent. It's just a question. If you quantify the uncertainty and propagate it, then you look at the results, and then you see where you can make the conclusion or not. Some of the, let's say, for example, drug development or activities, even if you have a really poor data, but they're so strong and so manifestful, you can even, the bad model can see the impact. Obviously, when we are talking about very refined therapeutics, which, really targeting specific cohort of people, then, you need to worry about, what is the error bar, and, because it definitely needs to target, in the right place, in the right manner. Okay, so so what are the major areas of data processing? So obviously, it's a data cleaning, data integration, data transformation, and data reduction. So, and obviously, we'll as we go right now, we'll talk a little bit about each single area or domain. So for the data cleaning, I already mentioned that, what are the steps to fill up some of the missing values, how to handle smooth or noisy data, identify, remove outliers, and resolve inconsistencies. There is a lot of techniques here. There is no some sort of unified approach, just take it as a granted and then just apply it all this work. It's in a sense, sometimes else as an art that, you, depending on the data and your domain of knowledge, build up an appropriate algorithm. There are certain common steps which, helps you to build some sort of a skeleton of that pipeline, and then you just fill up kind of a meat around that skeleton, depending on your data and depending on the issue you have with your data. Data integration, obviously integrating different assets. So those days you probably hear a lot of companies or big biopharma who is utilizing the concept of the big data and then building their lake of data and then trying to build drug discovery platform on those data lakes. Obviously, they will challenge the problem of agglomerating all those different sources of the data, different modalities, and then feed them into the framework of existing libraries. For example, TensorFlow, most of the method takes them in the form of the tensor. It could be just a vector, it could be a table, it could be a high-dimensional matrix, right? Therefore, the name comes from the tensor, because tensor is the most general object exists in the mathematics. So therefore, like, you can manipulate with the tensors of a different order, and then you can obviously, using sort of tensor operation, you can build, it's like multiplying two numbers, right? Or summing two numbers. You can actually work with the tensor. There are certain different operation you can perform with the tensor, but the bottom line that, agglomerating different information into the tensor structure and then combining them, you allow to agglomerate different modalities into the different method. Data transformation is also very important, and I'll show you a couple of methods and examples. And it's very important, especially when it comes to the fact that, sometimes different sources of the data, let's say you're trying to look at them, you're trying to compute unfolding temperature of the monoclonal antibodies in a specific assay. The pH, the salt of the assay, and then the sequence of the monoclonal antibodies. So obviously, those are 3 different types of information. So, and they have a different values, because the pH, it could be, let's say 6.5, and then the salt concentration could be, I don't know, 250 nanomoles, and then or moles, whatever you wanted to put it in as a designer. But the bottom line, the size of the numbers, and then also there are some amino acid which needs to be converted, because it's just collection of letters as a sequence, but you need to convert it into the representation which you can combine without the numbers. But the fact that you can use, let's say, one of the techniques called one-hot encoder, we'll talk a little bit about this in the case study. It will convert your sequence into the collection of 0 and 1s, right? But you still have a pH factor and you still have a salt concentration. So basically, normalization is one of the techniques which allows you to get each of the variable into something like undimensional scale. And then, in that undimensional scale, you remove the amplitude, and then you will take into account the distribution, right, around the mean value and then dispersion. And that allows you to, those transformed values after normalization can be used in some of the methods together, and that will lead to much better predictions instead of just, plug them numbers as is. So that transformation is a very important step. And we'll show a couple of examples I mentioned already. Data reduction, is it something, obviously very important, especially when we are talking about understanding certain features and then using some of the coarse representation of your study. Obviously, data reduction is an old concept which is still valid, but it was obviously from bottom to up type of data processing. So and the reason for this one was obviously constraints on the computational resources you can put in to analyze the raw data itself. One could argue those days that, you actually can allocate it, let's say somewhere in the cloud, enough computational resources, and you don't need to do the data reductions. Because while you're doing the data reductions, some of the data allows you perfectly in a statistical method reduce with very, very good accuracy to low representations and effectively embedded your high dimensional data into the low dimension space, and then do the analysis, which will be, of course, faster. But for some of the data, it's a tough call whether you truncate and what you keep it as your core features of the system and what you remove. And with the current situation of doing a lot of cloud computing and relatively cheap cost of the computational resources, you can, trying to tackle, as is without any dimension reduction. But it still need to remember, because it's also helpful to sometimes perform the feature importance analysis and to identify the primary target or a list of the highly likely target for your problem of interest. So for that reason, let's start with the data cleaning. Again, since most of the people are no voice or beginners, we have maybe a few with intermediate level, but I'm pretty sure you kind of intuitively see where is the problem. So if you have a data and then some of the values are missing, or you have a number of outliers or noisy data or some inconsistent data, that's something you need to address, because otherwise, when you're trying to build, let's say, regression or classification, yeah, the model itself will be very confused, and the error, basically minimization across all the observation, would be very difficult to perform because the data itself is ill-posed. So obviously there are many techniques how to address each of this, as already mentioned. Again, that's probably not the most important for today for our training course, but the simplest way, of course, is removing them. And if you have enough data and it doesn't affect, a majority or like a largest representation in your data, then you can just simply safely remove. But if you, after removing all the kind of bad values, still end up with a very small data set, which is, very hard to use to build a reliable model, then you need to look at some other alternative strategy, how to keep those data, maybe adjust them. And then in case, some of the, for example, reinforced machine learning models can be used to actually trying to find the missing values such that, subject to several iterations, that you still be able to honor other observations, right? And then, minimally deviated from observation which belongs to that missing, to the sample which has the missing values. And then you will stipulate it that, the missing value would be of that order. And that will allow you, of course, it's a synthetically generated value. We don't know whether it is true or not, but, you could assign the error bar to this missing value, because obviously the error bar could be built from the biophysical considerations. So let's say if you know that in which range that particular value could be, and then what you can do once you send the error bar to that particular value, you could do the multiple realization of different values in that particular interval, giving the median value which was obtained via reinforced learning. And you will have multiple outcomes of your machine learning model. It's not bad because, multiple outcomes leads to certain statistics, and then you can make analysis of that statistics and make certain hypotheses what would be the observation subject to the given distribution. Furthermore, you can make analysis of what is the confidence interval on that observation or that claim, and you can actually look at power analysis. You look at the what we call the power of that statistical hypothesis testing, giving the distribution, and you can make a lot of analysis out of that. So therefore, like, despite the fact that there was missing value, we assign certain value and then assign some error bar towards this one. We were able to move further down and generated some of the hypotheses and actually test them. And that applies to the noisy data. Of course, the noise can be handled and analyzed, and then some of the inconsistency data, which, the main message what I'm trying to say is that, before you kind of remove all the bad data, and if it is okay to do and you still have a sufficient amount of data left, that's fine. But, if you will be really facing the situation that, removing all the points will end up having no points or very small amount of points, then I would recommend you to trying to populate those missing values, error bar or the level of potential change of those variables, and then propagate that confidence or error bar towards the predictions, and then analyzes what the potential outcomes could be subject to the uncertainty and the missing value. So this is we already roughly discussed. Obviously there could be a different source of the missing value. So I don't think so even it is, very, very comprehensive in terms of describing all the source of the potential or reasons of the potential missing value. Sorry. But the bottom line, there could be some missing value, and we know that sometimes, you're doing, some of the trials with the animals, you are able to extract some of the information, but then something happens with the animals, you won't be able to extract the information. So therefore, like some of your longitudinal study may be not complete, etc. So for that reason, it's important to keep in mind that it happens very often, but we shouldn't be afraid of that, and we have some of the ideas and strategy how to mitigate those situations and still allow you to perform the machine learning and AI on the data. Okay, so I already mentioned a few steps what needs to be done. Obviously, ignoring the missing data is one of them. Filling the missing value manually. It's something, if you know, and you can just populate it, or you can build, some of the machine learning activity will populate and actually identify what the machine learning fits as the best value if some of the uncertainty interval assigned to this missing value. Obviously, we can use some of the global constant. You can always put a 0 to the missing value, but usually those are a brute force approach when you just assign one value without any reason is always very tricky because it potentially could harm a lot of analysis and leads to very misleading results and conclusions. Obviously, when it comes to the missing of the attribute, so then it's just we need to populate the attribute, and if it is a discrete categorical value, then I would try to use the machine learning techniques to populate that missing value, and then assign the probability how confident we are of that missing value across all other values in that categorical list. It's also possible. It will obviously takes a little bit time to run several times or several iterations of the machine learning, but you still will be able to identify some of the possible realizations and use those possible realizations to make the predictions and then see what will be the distribution of predictions, giving you initial starting point. Obviously, it could be some other approaches, including some of the hypotheses based on the Bayesian formula or some other correlation estimates. It's always sometimes people use them to populate the data and then make the analysis. Again, if you're trying to use any of the correlations or models, trying to quantify what is the uncertainty associated to this approach, and then trying to propagate that uncertainty to the final stage of the results. So that would be my recommendation and the approach how to handle the data. Okay, what about the noisy data? So in our case, noise could be very different compared to, let's say, automotive industry or spacecraft engineering domain. Noise could be biological variability. It could be noise coming from instrument. It could be some contaminant which comes into the assay. Things could be very different. So for that reason, obviously, we could use some of the model, which is, will take into account the modeling of the noise. You can actually build the machine learning model to predict the noise. That's in our approach, valid approach, and I've seen some of the pipeline where the people use the machine learning to approximate the noise. But the bottom line, again, if it's the case of the data, then your final result should not be just only one concrete prediction, but it should be a prediction with the confidence interval, because subject to the noise, you may be they are here, so therefore you need just to make sure that if you are doing classification, it always has to come up with the probability of belief, or if you are doing regression, it has to always come up with the error bar assigned to your predicted value. What are the other data problems which requires the data cleaning? Obviously, we already talked a little bit about the duplicate records. Some of the machine learning models actually would be happily taking the data and then running them and wouldn't even notice that there was a duplicate. It will provide reasonable results. For some of the method, it's essential to remove because they will lead to some of the matrices embedded into the mathematical description of the method, which needs to be inverted, and there will be a ill-conditioned matrix, and therefore, it's essential to remove this. Otherwise, you can maybe even have a crash or very lengthy process towards reaching the convergence while you're training your model. Obviously, incomplete data inconsistency we already discussed and addressed. So it's important aspect we need to take them into account, if possible, to resolve them in a kind of domain knowledge level, so that we assign certain values and some of the uncertainty associated with those values. So in our way of handling noisy data, but also actually not only noisy data, but doing regression in general, it's a binning method. So because if you know that we have some of the data points which could be in a certain range, so then we can, let's say, binding affinity split in the different intervals, let's say in a certain step, and then all the let's say monoclonal antibodies come into different beans, right, in terms of the binding affinity. Then instead of actually really regress and predict the value, we'll just predict to which bean it belongs. So effectively you're already some sort of trying to incorporate the error bar, because you're saying like my prediction is some sort of middle point of that bean plus minus standard deviation, which allows you to capture the lower edge of the bean and the upper edge of the bean. So, but it's only partially true, because when you do the classification, so you also have a possibility that that particular candidate can be in other classes or beans. So therefore, what usually you end up having is a prediction of the bean plus the error bar, which potentially may cover a few beans. You still will have that information to see like where potentially it could be. So it could be just one bean below or one bean above. So things like that you can obtain as analysis and uncertainty quantification, and it's always useful results. And then the case study we are going to look at today, which is related to the activity of CFI protein. So you will see that that information is important because it allows you to position different candidates in a different beans, and then you look at the error bar and you effectively select late in the study the most highly likely candidate with the increased activity. That's what we're interested in. And then you go to the experimental validation stage and you test those candidates and see whether your in silico prediction to provide a reasonable candidate or not. Clustering is also effectively identifying the samples which has the same signature despite the presence of the noise. It's not just basically one-dimensional binning, right? But it's actually more highly dimensional binning in a sense, because the cluster is a more general concept compared just one-dimensional axis, right? Even if you bin in two axes, it still will be in the squares, and clusters in a more general shape could be. So therefore, it's more, of course, powerful, but limitations are here is that depending on the method you use, some of the method obviously will generate different clusters depending what is your starting point, and if your data is really noisy, it could lead to some sort of stability issue in terms of identifying the most stable classes, which a number of clusters, and then distribution along those clusters. That's an issue because we don't know upfront how many clusters we should expect in our data, and therefore it's going to be a very lengthy and tedious process to identify the right method, the stable number of clusters, and then cluster all the data into those clusters. Of course, it could be semi-automated method in a sense. You could do semi-automated method of clustering when you change the number of clusters and you iterate it till you find appropriate number of clusters, with the multiple realization and different starting point, but roughly will end up to the same solution after all. And then you can say like, okay, I found the center of my clusters, they looks very stable, and at the same time I found the correct and stable number of clusters, and then all my data can be clustered into this one. That could be one of the semi-automated method. It could be some other realization of the manual plus automated. So obviously there is a huge variety of the method here, and the most important is to remember the concept that you can run the simple, very simple KNN type of cluster, and then define and see like how the data behaves. And if you see that it's already providing a reasonable solution, then you could use this one. If you see there's some of the problems coming up, then you can look for other alternative methods, the particular addressing the problem you observing with your data. And of course, the regression, something which will require some of them model for the noise, but it's still possible. And again, I already mentioned that the model itself could be developed using the machine learning, but most of the time you make certain assumption about your noise distribution or certain characteristics, and you perform the analysis of the data subject to this assumption. Okay, what about the data integration? So again, I already touched base on that subject. It's very important, especially in a current environment when you have all sorts of information. So most of the time, you do several levels of machine learning. You do the machine learning while you're processing, let's say, single cell data, right? And you're trying to identify different cell types, annotate them, identify different biomarkers responsible for the particular cell lineage, etc. It could be some other data, proteomics, it could be some other health data. There is a lot of data coming up which is associated with the patient. The question is, how would you combine this data? And then, the question, what is your ultimate goal? Do you do like classification? Do you do regression? Do you do deep learning? All those aspects will impact the way how you need to integrate the data. There are multiple ways. Again, remember that the most kind of powerful and at the same time one of the key elements to remember when you integrated the data, that each source of the data or type of data you firstly convert to tensor, and then you do the tensor manipulation, and you can design a new tensor result, so the, let's say, tensor product or some other operation which allows you to build high dimensional object which you can fit in a lot of methods, and then get results, machine learning trained, and then make the prediction. So while you're doing integration, it's also important to remember about consistency, right? So that, we're trying to eliminate that as early as possible. We also need to make sure that, we, especially when we have situation when they're trying to merge the data in terms of the cohort of the patient for the different type of disease, or even different, let's say, cancer form. It's important that, we do have a match in terms of the final outcome or ID levels, so that, we don't have any mismatch in terms of the cohorts. Otherwise, the data will be confused, resulting to the confusion machine learning and ill-posed predictions. So how do we resolve usually conflict when the data comes up? If you have conflicts on the identifying level or labeling level, so we usually same attributes or same data, we just basically check the consistency across the attributes, and we look at how we can eliminate those inconsistencies. Let's say if inconsistency comes on the unit level, so then obviously you need to convert everything to 1 units, or you need to perform like normalization, which obviously will eliminate of units all, because all the resulting data will be normalized and dimensionless. And that's usually what is recommended, just to avoid any confusion or any misleading results later on. One important aspect as well is basically similarity of the data, and where you need to redundant some of the entries. Again, for some of the method, it's probably not a bottleneck, and it happily can take the same repetition, right? Maybe you can use even some method to say, like, if what would be my some sort of machine learning model which will pass some sort of in between two observations, which is slightly different. However, it's coming from the same patient, but maybe done in slightly different moments in time. But usually people do apply some sort of correlation analysis, and they're trying to understand what is the correlation between various samples, and then do the cleaning of the data on that level, which is it doesn't hurt to do it. Again, when you do these things, make sure that you don't throw out a lot of useful information. So the question is like, what will be your cutoff in terms of the correlation coefficient? When do you drop some of the data, when you keep some of the data? So what I would suggest, again, do multiple realization for the different cutoff or threshold level, and then use those multiple simulation later on into the prediction stage. So effectively, you kind of propagate the uncertainty of the correlation analysis to your final prediction results. and then look into those multiple realizations, and the final stage you would see what would be potentially the distribution of your outcome, and you can make some sort of claim about most commonly observed outcome based on your data. Okay, so data transformation. So we talked already about this. Obviously, sometimes there are a number of methods which is well known and defined. You know, some of them are related to the smoothing, which removes some of the noise, which is, you know, very good to do, especially when you have a very noisy data and you just, you know, pre-smooth it using one of the filter. There are many techniques, waveform inverse analysis, filters, and there's a bunch of the different methods available in different platforms, in R, in Python, in MATLAB, depending what is the choice of your developing platform. You could perform that smooth. You can remove some of the noise. So the issue with the removing the noise is that you have to be very careful that you don't smooth out the real data. And if your biophysical processes is noisy by its nature, right? So you need to differentiate, you know, that noisy situation versus real noise coming from the instrument or any other sources which should not be present in the data. So again, aggregation is another step of the data transformation. So in a sense, summarization and data cube construction, which allow you to build that potential hypercube of different realization of your data, and then, you know, simulate for each of the point of your data cube, and then obtain the results and building the distribution of results. I think, you know, it's a very valuable approach. It allows you to see what is the uncertainty in your data propagating into the results. It also allows you to capture the different type of the noise present in the system. So overall, it just gives you ability to stretch and stress your model subject to different perturbation of one of your variables. Generalization, obviously, moving, you know, and building heuristic climate concept is nice and allows you to transform data into the low dimensional space and utilizing this information and building the model. But again, it's something maybe not so intuitive and naive. So maybe you just, you know, better to start doing the machine learning on your original data before you actually start building any hierarchical model or doing generalization. So normalization, we already talked about z-score normalization. We talked about it could be min-max normalization or normalization by decimal scaling. So those are the most commonly used techniques. Again, the bottom line, the idea is that you're trying to eliminate the factor of the scale and then trying to incorporate, kind of map everything to the same axis, you know, given the fact that all the data could be very different in their nature. And of course, new attribute constructor from the given ones, that's something important, especially when we see that sometimes it's very difficult to, let's say, classify the model in a multi-class type of problem. Say you have a 10 cohort and you need to build a machine model which will be basically distribute the data into all the 10 in one. So then making the unification, or even, let's say, doing the binning in a more heterogeneous way, not a uniform step, but, you know, let's say you have a lot of bins at the beginning of your binding affinity data, and then, you know, less at the top, which basically allows you to capture less, you know, highly, let's say, binding, you know, site into the coarse bin, and then more refined at the bottom. But the bottom line is that it depends, you know, where you want to make your focus. If you're focusing, let's say, at the bottom spectrum of your binding affinity, you'll refine it there. If you're more interested in the top, then you just will make one coarse bin at the bottom and then more bins at the top. But all those things will allow you to actually get less classes or less bins, and then you can much easier and better to classify and build, you know, the machine learning and make a prediction. So that's the most important. So this is how the example looks like, for example, for transformation. So we're going to use the z-score normalization in the case study I'm going to show you later on. So maybe in 1 hour or so. So you'll see the example of this one. But it shows you now what does it actually look like, when you do the normalization. So it's not a rocket science. So you see that's a pretty straightforward expression, but it allows you to get, much better quality input data when you do the machine learning construction. So maybe that's just a quick example again, but I don't think so. We need to spend too much time here. That just show like, if you know your mean and max, and you know your new mix and max, you can always scale up, your original data in a variable new into the new one, just basically applying all these parameters into the formula and then obtain the new value, which will be a mean max normalized formula in a new scale. So the data reductions, we already talked, and the reason why we need to do it sometimes. But again, maybe that strategy in nowadays when the computational resources is less of a concern. So it's maybe not the most high priority, especially like sometimes you're really interesting to keep all the data in place and see what would be outcome versus when you do pre-processing and you build the course data and then you do the analysis. The only way sometimes is basically time limitation, because if you want to obtain some results very quickly, just to get a sense, where you're moving on that, it makes sense to reduce the data, make the problem smaller, solve the problem faster, get the results, see where you're shooting your project or your analysis in the right direction. Then you build the confidence in your pipeline, you obtain, much deeper raw data, and then you use already a machine learning on original raw data without doing any reduction. That could be one of the potential approach for applying machine learning. Data compression. So it may be interesting. So in terms of utilizing that concept inside the machine learning, not for the data processing itself. Like I said, you can take the original large data, you can build that large tensor, but it will be some sort of compression involved during the training of your machine learning model, which is basically compression step will be part of the machine learning algorithm. And the reason for this one, because you wanted to train the model fast, and then you have to do some of the compression and some of the reduction of your data, be able to meet certain reasonable training time. So obviously, it has been a lot in terms of audio, video, and image compression because of the mainly storage aspects. And that's not something we're using actively during our activities when it comes to the life science problem. But some of the algorithm and methods is very useful, especially in the case of the CFI protein case study. You will see how we compress the data there, and what you can actually gain out of the compression method at the point of actually building the set of the potential mutation, identifying the residue, which is sensitive towards the mutation and will lead to change of the CFI protein activity. So principal component analysis is another way of reducing your data. Obviously, you're probably all familiar with this one. It's one of the key elements in assessing the quality of the data, and then get a little bit sense what is happening there. Again, remember this one, it's a useful tool. Doesn't hurt to perform the analysis to see how things are distributed and see like where the outliers, etc. So always very useful tool to perform with the data before you actually do the analysis. Again, identifying the principal directions and then see what is the magnitude of the principal direction. The eigenvalue corresponding to this direction is an important metric and measure the impact of that particular vector towards the description of your data. Again, maybe a few words about reduction in terms of the parametrics and non-parametrics. If you're able to fit, some of your data with the models, let's say, when you do, let's say, modeling of the noise, sometimes we assume it's a white noise with certain characteristics, and then you do the simulation. So it's a kind of related to parametric studies. But if you are not able to assume any models, so obviously you can do non-parametric models. You just basically do random sampling, assign it to a value of that model, and then generated the different realization of the model depending on your sampling. That's also a valid approach, especially if you're not able to confidently enough assume some of the models for your data. Yes, now we are coming to regression analysis, and assuming that we already clean up the data, it's in a reasonable state, and then things what we can do. And sometimes as a first step is just do simple linear regression or multi-regressions, but still linear multi-regression analysis or log linear regression analysis to understand some of the basic characteristics. Let's say log linear model allows you to answer some of the questions where the distribution is normal, not normal, some other things. You can also be able to understand, what are the variables more important, especially looking at the coefficient of yours linear regression analysis, and also look into the error which you generated. And you could actually see where you need to do any more advanced fitting of your data using, let's say, neural networks or highly non-linear function for your regression model, or you can use some other method. But the bottom line, so those are first initial steps. Sometimes not bad to perform, just to understand, where you need to move, to more complex realization, more complex model to fit your data. Obviously, you're probably well familiar with linear regression analysis. We're not going to spend too much, but what I just want to point it out here is that some of the informations in some of the steps of the linear regression analysis, they do replicate in a neural network, right? So because at the end of the day, a neural network is some sort of functional form which maps your data into your desirable observation, especially when we're talking about the supervised machine learning, and identifying some of the characteristics of each of the layer of the neural networks as effectively finding the parameters, which in terms of the linear regression, you see those parameters in the model. And then the whole game is about how quickly you can find the reliable set of the parameters for each of the layer of neural network, for example, which minimizing your error. So in some of the questions, you could have a multiple solution to this problem, because minimization of the error giving a very complex shape of your function may result in multiple local minima for the error surface or loss function surface. And therefore, depending where you start, and sometimes you see that, because some of the algorithm has a random generating starting point, you all this time can see slightly different results in terms of your neural network and some of the prediction results as well. You always need to keep it in mind, so that where the source of this randomness coming from, and why do you see the different realization, despite the fact you haven't changed anything, you just run the same pipeline, the same model. Okay, so we talked already about the histogram, so we can skip. We probably already talked a little bit about the clustering, so I don't think so too much to add here. There are obviously different type of the clustering method. Again, we can spend an hour talking about the different clusters, limitations and limits, so we simply don't have a time. But the bottom line, clustering is used in many aspects. It's unsupervised learning as well as for the data cleaning, can be used to understanding your data. So it's a very handy concept. So keep this in mind, but at the same time, keep in mind that some of the methods really unstable. Again, depending on the number of clusters you're looking at, they can be, very different, distributed, and even as well as unstable. If you run even the same number of clusters, but you have a different starting point, you could lead to a very different solution in terms of distribution of your samples across those clusters. So that's very important to remember, and maybe, classify them, keeping all the possible organization, and then on the top of those possible organization, build the predictions, and then see what is the potential outcome. So which, will be subject to your original choice of the clusters and the distribution, and what would be the outcome allows you to build answers with some of the probability distribution, probability belief in your answer. Sampling is again, we already touched base on this one, especially that would be very important in a situation. Imagine that you have a problem to classify the patient in a 2 cohort, and one of the cohort is really small size. You could have, let's say, cancer classification problem. You have a lot of samples with the negative status, let's say 1,000, and you have only 10 with the positive. So obviously, if you just keep 10 versus 1,000, it's really disbalanced data. And and it's very important because if you build a machine learning just using 10 and 1,000, the machine learning can pick up and say, like, let's just classify all the time that all people are negative, and we will just make it this 1% of error, because we know that based on this data 1,000 is negative and only 10 is positive, and it doesn't kind of reflect what you wanted to achieve. So therefore, for this reason, there are various techniques how to address undersample class. One of them is a sampling. So what you can do, you can select randomly 10 samples out of 1,000 and then combine those 10 samples with the 10 positive samples. In that case, you have 10 by 10. In a sense, the situation, you have 10 in the negative case and a 10 in the positive. You can build machine learning, although the 10 maybe on itself is a small sample size, but at least you remove the the issue of disbalance between two different cohorts, and that's one of the good application of the sampling. There are many other situations where you can do the sampling, like one I mentioned previously, that you can sampling if you don't know the model, any parametric model behind some of the parameters, which is very noisy. So in that case, you can sample out of your data some of the values and use this later on to predict, and then you just look at the possibility of your predictions and build the final results with the probability of the belief based on the distribution of possible answers. So again, regression is just basically what we're trying to do is building the language process for the point. We already talked about this, no much time, there's no need to say here. So what we usually look at, most of the time we are fitting the data subject to noise. Again, we talked already about, so that we handle this one, we can assume some of the model for the noise. It could be, normal distribution with mean zero and unknown variance. It could be some other distributions. And the bottom line that, we're trying to find the probability of predicting our observation Y subject to the coefficients and given data. And then look at the possible distribution, and we'll be able to say what would be the possible outcome. So we could imply different forms of the linear regression. One is the Bayesian one. Let's again not spend too much time on that, but what I'm trying to just make the point here is that again, similar concept will appear in a different structure of neural networks, because a lot of concept actually was born from the simple regression analysis and nicely penetrated towards a neural network in terms of the quantifying the error, in terms of finding, different optimization, the search path, etc. So we'll talk about this. So eventually we'll always, trying to maximize some of the loss function. The loss and shape of the function could be very different. One of them could be just product of a different probability of observing certain observation subject to the weight and given parameters. It could be some other forms of the loss functions. In some of the methods, the shape of the loss functions you can select as a user, and that's something, as I mentioned, worth trying if you don't happy with the default value. And also you can optimize, different search strategy for your loss function. Sometimes it also helps to get the better machine learning model for your data. So obviously you can demonstrate it in a nice and elegant way that, maximizing one type of loss function, it can actually show that it's equivalent to minimizing some of the square methods which appears in a simple linear regression analysis. And that's something, what I was already mentioned, that, some of the concept and some of the generalization which appears in machine learning, they all connected to, in some way, logically to the conventional linear regression analysis. And actually they reduced even to this one if you have a very simple situation, and you can demonstrate it that some of the model do actually represent, or reproduce the linear regression analysis and provide the same answers. So I think it's one of the new type of test, or sanity check, as you wish, for more complex neural network model you are trying to build. So they always need to take into account that they can reproduce the previous results. Again, we can skip this one, maybe skip this one. So maybe if you points I would like to mention that in Bayesian study stats analysis, we basically have ended up with probability of distribution of W, which is basically what I was trying to say, that sometimes you wouldn't be just able to find only one W, so your weights or your coefficient in your analysis or weights of your neural network, but it could be distribution because of a different uncertainty coming up, depending on what is your data, right? And obviously, looking at this distribution, you will be able to make certain conclusion about what will be the most common observed machine learning or basically weights, and then make out of that the prediction about the most commonly observed outcome of the prediction step. And that would be a nice incorporation of your uncertainty, which comes from the data towards the machine learning, and then finally it will reflect in the results from the machine learning. The same story about the multi-variable regression analysis. We'll just quickly skip this one and just summarize for the sake of the time, because I know we're kind of a little bit late and I want to spend more time on the case study. So obviously the data preparation analysis cleaning is important. Remember about garbage in, garbage out. So however, obviously life is not perfect. Sometimes you cannot just simply remove bad points, because otherwise you'll end up with having no points. It's important to see where we can improve or assign certain values to the missing point, keeping the consistency with the data, but at the same time assign the uncertainty to those values and propagate those results to final results, and then build the distribution of possible outcome and analyze those possible outcomes to make the final conclusion, what would be a statistically more significant observation. Obviously, integrating the data is important. Normalization is a very important aspect. So we talked about reduction. In some cases, it became very important. Maybe sometimes it can be dropped, but it's still important to remember all those concepts in mind, and trying to be open-minded in a sense, like, if somebody offers you some of the pipeline and they assume certain things, you should always challenge them to see, like, well, why did they drop some of the steps? Is there a particular explanation, or it's just because they didn't think about that step in the first place, and just overlook the potential impact of that step on the final results. All right, okay, that's it about the data wrangling, which is the essential part of the machine learning.